Hello students, in this video we are going to study about Human Genome Project, that is the last topic of the chapter 6, Molecular Basis of Inheritance. Human Genome Project is also known as the mega project. So the question arises, why it is called as a mega project? Human Genome Project is called as a mega project because it involves lots of money in the execution of the project. Various advanced techniques has been used. Numerous computers were involved for the storing of the data that has been used for the sequencing, and the scientists, an ample amount of scientists were involved for the success of this project. Now let us see the cost, how the cost affects this project. As we know that the haploid content of the human DNA is approximately 3.3 into 10 to the power 9 base pairs, and if the cost of sequencing of a single base pair is around $3, so the overall cost for the human gene or the human genome will comes around approximately $9 billion. Now let us talk about the storage of the data. Since the haploid content of DNA is 3.3 into 10 to the power 9 base pairs, so the storing of the data or the sequences that has been done due to Human Genome Project also requires to be done. If the sequences are stored in a book in a typed form, then we need a book which has at least a page with 1000 letters in a single page, and similar pages we need approximately 1000. So if 1000 pages are required for 1000 letters in a single page for the storing of total information of a single cell, requires approximately 3300 books. So the storage becomes the great havoc. So in order to store the data of the sequenced DNA, as we know that human is a multicellular animal, so a numerous amount or million of cells are there in human cell. So the enormous data needs to be generated, which will be associated with the development of a new era of biology that leads to a development of bioinformatics. Now let us see the Human Genome Project. It is a 13-year project which starts from 1990 and completed in around 2003. It is coordinated by US Department of Energy and National Institute of Health. The aim of the Human Genome Project, was to study about the effects of DNA variation among individuals that lead to revolutionary new ways to diagnose, treat, and prevent numerous disorders that somehow affect the human beings. Apart from sequencing the human genome, there has been other non-human models that has been sequenced. For example, that of C. elegans. It's a free-living, non-pathogenic nematode. Another one is Drosophila melanogaster, the fruit fly, and the plants that has been sequenced are rice and Arabidopsis. Now coming to goals of Human Genome Project. It defines the identification of approximately 20,000 to 25,000 genomes in human DNA. Human Genome Project helps to determine the sequences of approximately 3 billion chemical bases that helps to make up the genome of human DNA. Apart from the sequencing, the storage of information has been done in the form of databases by the process of bioinformatics. It helps in the improvement of tools for the analysis of data, and it also involves the transfer of related technologies to other sectors like that of industries. Human Genome Project also helps to address ELSI, that is, ethical, legal, and social issues that may arise due to Human Genome Project. The methodologies that are used in the Human Genome Project is expressed sequence tags. It is the technique by which the identification of all genes are done that are expressed as RNA. Sequences of all the genes are done that are expressed as RNA, and those genes or the mRNAs are called as ESTs, that is expressed sequence tags. Another methodology used in Human Genome Project apart from EST is sequence annotation. In this technique, the whole genome is sequenced irrespective of their coding and non-coding regions, and later these regions were annotated or assigned the different regions with functions. Now coming to the process, how the process has been carried out in Human Genome Project. First of all, the DNA is isolated from the cell, which is then broken down randomly into fragments with the help of a molecular scissor called restriction endonucleases. These fragments of DNA are then inserted into vectors, the cloning vectors, that is BAC or YAC. Let us see the full form of BAC. BAC is bacterial artificial chromosome. YAC is yeast artificial chromosome. It involves the insert of DNA of approximately less than 300 kb, and YAC involves the insert of DNA from the size of around 200 to 2000 kb. And these fragments are then closed into the respective host. Bacterial artificial chromosomes are cloned into bacteria, that is E. coli, and YAC will be cloned into the host yeast, that is Saccharomyces cerevisiae. After the insertion, the multiple copies of DNA are produced by the technique polymerase chain reaction. These fragments are then sequenced as an annotated DNA sequence, and the sequence are then arranged on the basis of overlapping regions. These necessitates the generation of overlapping fragments for sequencing. The computerized programs were then used to align the overlapping sequences, and at last these sequences were then annotated and assigned to different chromosomes, leading to the sequencing of all the human chromosomes, that is the 24 chromosomes, 22 autosomes, and X chromosome and Y chromosome. The last chromosome that has been sequenced was the chromosome 1, and it has been done in May 2006. Thus, the Human Genome Project has been accomplished with the help of polymorphism, that is variation in the genetic level, in microsatellites using restriction endonucleases or the recognition sites. The genetic and physical maps of the genome was prepared, and thus the Human Genome Project has been accomplished. Now coming to the salient features of Human Genome Project. Human Genome Project involves the sequencing of human genome. Therefore, it contains approximately 3.1647 billion base pairs, with the average size of gene having 3000 base pairs. The human genome consists almost 30,000 genes within which almost 99% of the nucleotide bases are exactly same in all the people. Next is the largest gene that is present on X chromosome have the size approximately 2.4 billion base pairs, and it is responsible for the disorder Duchenne muscular dystrophy. Other gene that has the size of approximately less than 10 kilobases are the gene that codes for beta globulin chain and the insulin gene. Now, the maximum number of genes are present on chromosome number 1 with 2968 genes, and the minimum number of genes are present on chromosome number Y, that is 231 genes. The functions of over 50% of the discovered genes is unknown, and less than 2% of the genome represents the structural genes that codes for proteins. Only 0.1% genome with some 3.2 million nucleotides results into the variation that is observed in human beings. At about 1.4 million locations, there is a presence of SNPs, that is single nucleotide polymorphism, which plays a potent role to find the location of the chromosome that are associated with the diseases, and these are then sequenced and traced for the human history. Now, the largest amount of human genome, or the largest portion of the human genome, contains repeated or repetitive sequences, and approximately 1 million copies contains 5 to 8 base pair of repeated sequences that are clustered around the centromeres that are present at the end of the chromosome, and these chromosomes or the DNA sequences are called as junk DNA. Now, coming to the applications of Human Genome Project, it helps in the identification of disorders. That is, more than around 1200 genes are responsible for numerous disorders like cardiovascular, endocrine, or neurological diseases or disorders, which can be assessed with the help of Human Genome Project. It helps in the detection of cancers, where efforts are in progress to determine the genes that helps to change the cancerous cells to normal cells. Healthcare has also been benefited by Human Genome Project, where the prospects of healthier living, genetically modified diets, and designer drugs are evolved. Apart from them, numerous tissues, organs, and tumors are also analyzed to cause the effect using Human Genome Project. That's all with the Human Genome Project. Hope you like this video. Thank you.